Welcome back, students. In today's class, we are going to study about cell division, which is an essential life process. And one type of cell division we will be studying today is mitosis. So, let's begin. Cell division is one of the very important properties of cells and living organism. Due to this property only, a new organism is formed from the existing one. A multicellular organism grows up and an emaciated body can be restored. Emaciated body means very thin body. So to gain muscles or to put up the weight, we need the process of cell division. There are two types of cell division as mitosis and meiosis. Mitosis occurs in somatic cell and stem cell of the body, whereas meiosis occurs in germ cell. Germ cell means reproductive cells. So mitosis occurs in all the cells except reproductive cells. Each cell has a nucleus. Besides, other cell organelles are also present. You all are very well aware of that, children. Let's study the cell division with the help of all the previous information that we have. Before going to the cell division, quickly we will recap chromosomes. Chromosome structure you have already studied in 9th standard. You all know that it's a dumbbell shape or X shape. Here centromere is there, which is kind of a you can say the middle point of it, and this is called as chromatoid. Before any type of cell division, the cell doubles up its chromosome number present in the nucleus. That is, if the chromosome number is 2n, it is double up to 4n. Because when the cell gets divided, this chromosome numbers has to go each cell. Okay, so therefore when it doubles up, then each cell or each part gets the proper number of chromosomes. Now going to the first type of cell division, that is mitosis. Somatic cells and stem cell divide by mitosis. Mitosis is completed through two main steps. Those steps are karyokinesis. Karyo means nucleus and kinesis means division. So it's a nuclear division. And cytokinesis. Cyto means cytoplasm. So it's a cytoplasmic division. Now first we will study the first phase, that is karyokinesis, which is completed through 4 steps. Those are prophase, metaphase, anaphase and telophase. Let's study each phase in details. Going first to prophase. Now let's see exactly what is happening in prophase. Condensation of basically thin thread-like chromosome starts. Condensation means coming together. Chromosomes which are thin starts becoming thick. Due to this, they become short and thick and they start to appear along with their pair of sister chromatids. Sister chromatid means identical copies formed by DNA replication of a chromosome. Okay, so as you can see in both these diagrams, here we see the first phase. Here the condensation of chromosomes is being taken place. Okay, and here you can see that this is the structure of a animal cell, okay, or any kind of cell basically. So some organelles we can see here. Centriole duplicates and each centriole moves to the opposite pole of the cell. Here we can see the centriole, it also duplicates and then it moves to each pole of the cell. One may go here, 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 poles. Okay, so we will study further what is the use of the centrioles. Nuclear membrane and nucleolus starts to disappear. The nuclear membrane that is there and the nucleolus, that are the organelles, they also starts to disappear. So first phase, that is prophase. Moving further to metaphase. Now what is happening in metaphase? Nuclear membrane completely disappears in metaphase. Chromosome complete their condensation and becomes clearly visible along with their sister chromatids. As you can see in this diagram, what you can see here, the nuclear membrane is completely disappeared and we can very clearly see the chromosomes with their sister chromatids. Now see children, this red one and blue one, this is shown in different color, but they are identical copies. These are called as sister chromatids. Okay. All chromosomes are arranged parallel to the equatorial plane, that is central plane of the cell. This is the central plane of the cell and you can see that all the chromosomes are arranged equatorial to the parallel plane or central plane. Special type of flexible protein fibers which are called as spindle fibers. These are spindle fibers, as you can see in the diagram. The spindle fibers are there which are holding the chromosomes. These are formed between centromere of each chromosomes and both centrioles. So this end, these are the centrioles. Previous diagram we saw the centrioles duplicates. One goes at the top, one goes at the bottom. And this spindle fibers are holding the centromere. Centromere, as you can see here, it is the middle point, okay, or where the sister chromatids are attached with each other. And what you can say is the centriole and sister chromatids are held tightly or basically are being taken care by the spindle fiber. Here also you can see that these are arranged in the equatorial plane along with the help of spindle fiber and the centromere. Third phase is a anaphase. What is anaphase? In anaphase, centromere splits and thereby sister chromatids of each chromosome separate. Now see here in this diagram, what you can see here, same coming previous to blue and green. Now what you can see here is these are separated and have moved apart from each other. And where they are going? They are pulled apart in opposite directions with the help of spindle fibers. So with the help of spindle fibers, they are moving or to the opposite direction. Separate sister chromatids are called as daughter chromosomes. So these are called as labeled in the diagram, they are daughter chromosomes. Chromosomes being pulled appear like a bunch of bananas. Okay, so that is just, you can assume that it's a bunch of bananas. In this way, each set of chromosome reach at two opposite poles of the cell. Okay, so this is one pole of the cell, this is the opposite pole of the cell. So as we can see that each set of chromosome has now reached to the opposite pole of the cell. Even we can see in this diagram how the chromosomes have been pulled by the spindle fibers, are separated from each other and going to the opposite poles of the cell. Going to telophase, which is the last phase of this karyokinesis. The chromosomes which have reached at the opposite poles of the cell now start to decondense, due to which they again become thread-like thin and invisible. So first phase, condensation, coming together, becoming thick. Last phase, decondensation, going apart from each other and becoming thin. Nuclear membrane is formed around each set of chromosome reach at the pole. Okay, so we can see in this diagram the chromosomes have, now here we can see the formation of kind of two cells. Okay, the centromeres are there, complete nuclear envelope is there, decondensed chromosomes are there, and a contractile ring. Contractile ring means now the pulling will start so that they get separated from each other. Thus, two daughter nuclei are formed in each cell. So we can very clearly see the formation of two daughter nuclei. Nucleolus also appears in each daughter nucleus and spindle fiber completely disappear. So now the work of the spindle fiber is over. Okay, so spindle fibers are completely disappeared and very clearly we can see the nucleolus in two daughter cell. Okay, so here the end of the first phase, that is karyokinesis, and the cell moves to the second phase, that is cytokinesis. Now coming to cytokinesis. Now you can see from the diagram, the cytoplasm divides by cytokinesis and two new cells are formed which are called as daughter cells. So we can see very clearly the formation of two new cells. In this process, a notch is formed at the equatorial plane of the cell which deepens gradually and thereby two new cells are formed. So previous diagram we saw how the notch is formed. Here also we can see the formation of the notch and have been pulled apart and formation of two daughter cells. This is for the animal cell. Now even plant cell also divides the same way. So in case of plant cell, instead of the notch, a cell plate is formed exactly along the middle of the cell and thus cytokinesis is completed. Okay, so both the phases, that is karyokinesis, nuclear division, and cytokinesis, that is cytoplasmic divisions, are completed, and from one cell we get two daughter cells. Why mitosis is essential? Before that, this diagram is a quick recap of mitosis. What is happening is prophase, that is for condensation of fibers, then metaphase, that is separation or pulling apart of the fiber going to the pole with the help of spindle fibers. Then we have the anaphase, which shows the separation of chromosomes and moving towards the opposite and telophase, which shows the formation of two new daughter cells. And why it is essential? We have already studied. It is essential for the growth of the body. Besides, it is also necessary for restoration of emaciated body. That's a very thin body, wound healing, formation of blood cells. For all this, we need mitosis. Okay, so we have studied when a cell of the body is injured, after some time it again is restored because of the process of mitosis. Okay, and wherever there is a wound or any kind of particular kind of a wound healing in the body takes place also with the help of mitosis. Same thing in the plants when we pluck the flower. Okay, obviously when we pluck the flower, the plants also get injured. But again, by the process of mitosis, it restores the body. Okay, so the process of mitosis is very important for plant as well as animal body. So I hope you are very clear with what is mitosis. In the next session, we will study about the second type of cell division, that is meiosis. Thank you very much. Take care. God bless you.